Dear class 10, we'll continue chapter number 6, that is photosynthesis. Last time we talked about the definition of photosynthesis, components involved in the process called photosynthesis. The process of photosynthesis where we form two phases. They are light dependent phase and light independent phase. That all we did in the last sessions. Today or in this session we'll do experiments on photosynthesis. Okay, experiments on photosynthesis to confirm it or to prove that the different components like carbon dioxide, chlorophyll, sunlight are needed for photosynthesis. To prove that whether it is true or not, okay, to prove that we'll do the experiments today in this session. All right, so for all those experiments, two of the things that you have to do in the beginning itself, or two of the things that you have to know in the beginning itself, they are de-starching and to test a leaf for starch. These two things we need to know first. All right, so beginning with de-starching. Okay, so de-starching, what is de-starching? So de-starching is a process of removal of starch from the leaves. Okay, so to carry out the experiment, first of all we need to remove all the starch present in the leaves. Okay, so removal of the starch from the leaves is called de-starching. So how we do this? So we do this de-starching by keeping the plant which you have taken for the experiment, which I have considered for the experiment. Okay, that plant, by keeping that plant in a dark room or dark place for about 24 to 48 hours. That means 1 or 2 days, so that the leaves remove all the starch from there and store them in the different storage organs. So that is called de-starching, which means removal of starch. Okay, removal of starch is the de-starching. That means removal of starch from the leaf itself is de-starching. All right, so that is de-starching. So after de-starching, now what we have to know is that testing a leaf for starch. We are claiming that the plant is discharged or the leaf is discharged. Okay, that is our claim, whether it is true or not. For that you need to test it. So for testing, what you do is that take a leaf from that plant. Okay, take a leaf from that plant, the plant which you have already kept it in a dark place for 1 or 2 days. So from that plant, take a leaf, all right, and dip it in the boiling water for a minute. Okay, at least for 1 minute. Keep that plant or dip this leaf in boiling water for 1 minute so that the cells of this leaf are all killed. All right, then what you do is that then take a test tube here. Take a test tube here. Okay, this test tube containing methylated spirit here. Methylated spirit, as I have written out here. Okay, take a test tube containing methylated spirit here and dip this leaf here inside. All right, the leaf which is discharged is already put there in the test tube containing methylated spirit like this here. All right, and keep this test tube here in this position in the beaker. This is the beaker here. In this beaker here containing boiling water. This beaker filled with boiling water. That means this beaker is containing the water which is boiling. Okay, there you have to keep this test tube containing now the methylated spirit and the leaf. All right, then you have to keep boiling this till the leaf becomes pale white. Okay, you have to keep boiling it till the leaf becomes pale white. Alright, then after that, when it becomes pale white, then take it out. Take this out. So when it becomes pale white, that time the leaf is brittle. Okay, the leaf is now brittle. It is breakable. Okay, it is breakable. So it may not be, it may not be suitable for the taste. Now what you have to do is that you have to soften it. You have to soften the leaf by bathing it with the boiled water again. Okay, or the hot water. You dip this leaf again in the hot water. So that the leaf becomes now soft. Alright, so after softening the leaf, you spread that leaf in a dish. Okay, in a dish like that. Alright, in the dish, or you just spread it in a plate or dish like this. Then what you do is that you put some drops of iodine here. Iodine test. This is the reason why it is called iodine test. You require iodine to test whether the leaf is containing the starch or not. Alright, so now put some drops of iodine out here. Some drops of iodine here on this leaf. Alright, so what happens is that when you put some drops of iodine on it, the leaf turns to brown coloration. It turns to brown coloration. Why? Because the leaf has not contained any starch. Okay, why it is turning brown? Because the leaf has not contained any starch in it. Had the leaf contained starch in it, would have turned to blue-black. No, would have turned to blue-black. Alright, so had this leaf contained starch in it, the leaf would have turned to blue-black soon after the leaf comes in contact with iodine. But it is not happening. So why? Because there is no starch. So that now the leaf is turning to brown. Alright, so when it is turning to brown, then it is confirmed that the leaf is free of starch. So this is how you need to taste it. So after every experiment, you need to taste it by bringing the leaf in contact with iodine. If it is turning to blue-black, then obviously there is starch. But if it is not turning to blue-black, other than blue-black, suppose it is turning to brown, yes, it is not containing the starch. So that way, we need to make sure that it is discharged by testing it with the iodine test by this method. Experiment number 1, to show that chlorophyll is necessary for photosynthesis. As you all know that chlorophyll is very, very important for photosynthesis. Okay, and that's the reason why we say only those plants where chlorophyll is present, the photosynthesis is possible there. Alright, otherwise it is not possible. There are some plants like organisms like fungi, mushrooms and all, where photosynthesis is not possible because chlorophyll is not there. Alright, so to prove that, here what you have to do is that you need to consider a variegated plant. What is variegated plant? Plant containing multicolored leaves. Usually the leaves are green in color, but there are some plants where the leaves are multicolored. Okay, like this you see here. Okay, here is a leaf where you can see the multicolored leaf, where not only green portion is there, but there are white portions also there. So those plants which contain the leaves like this are called variegated plants. So consider a variegated plant for this experiment and keep this plant in a dark place for 1 or 2 days for discharging purpose. Okay, then after 1 or 2 days after discharging, what you do is that you just take a leaf. There are many leaves, but take one leaf or consider only one leaf for this experiment. Alright? Okay, so before taking one leaf out, before removing the leaf, first after discharging, keep the plant again in the sun so that the photosynthesis takes place there. Okay, so after some hours, you need to take one leaf. As I was telling you, just take one leaf from there, and now you just draw an outline of this leaf on a paper like that. On a paper, draw an outline of this leaf here like that on the paper, and mark the different colored regions, like this color is there, this region of this leaf is green, mark it green. All right, and the other region where it is white, let us say, so mark it white. Okay, so after marking it, now go for the test, which is called iodine test, which I have done, which I have come to know, how we have to test it, that you all have already come to know. Go for the test. So for testing, okay, or after the test, what you can confirm out here is that those portions of the leaf which are green, you find turning to blue black. Those portions which are green, you see turning to blue black, confirming that there is starch. There is what? Starch formed. Why? Because chlorophyll was there. And the other portions which were white, okay, which were white like this, you see turning to brown, confirming that there is no starch. Okay, so from this experiment, what you understand is that only those regions, only those portions of the leaves, or only those portions of the plant where chlorophyll is present, the photosynthesis is present. Photosynthesis is possible there. That means how important the chlorophyll is there for photosynthesis, we can understand from this experiment. Now experiment number 2, to show that sunlight is necessary for photosynthesis. Another important factor for photosynthesis is sunlight. Okay, if there is no sunlight, photosynthesis is not possible. That's why we have already discussed that only during the daytime photosynthesis takes place. To prove that, let us carry out this experiment. Alright, for this what you do is that take a plant which is discharged. That means you have already kept that plant in a dark place for 1 or 2 days for discharging purpose. Alright, then what you do is that you just after discharging, consider one of the leaves. Consider one of the leaves and cover the middle portions of that leaf with black paper or tape like this, as I have drawn out here. Okay, this middle portions has been covered with the black paper or tape. Alright, okay, then just leave this discharged plant, which is end where one of the leaves has been covered like this, out in the sun for some hours. Alright, then after some hours, remove that covered leaf only. Okay, remove the covered leaf only and take it for the taste, starch test, whether the starch is present there or not, in which portions of the leaf the starch is there, or in which portion there is no starch. Let us take it for the taste. Now experiment number 3, we have now to show that carbon dioxide is necessary for photosynthesis. Another important component of photosynthesis is carbon dioxide, alright, which the plants get from atmosphere. Okay, if there is no carbon dioxide, means photosynthesis is not possible. To prove that, what you do is that you just take a plant which is to be de-starched again, keeping this in a dark place for about 1 or 2 days. Okay, so after that, you see, take a twig containing some, leaves out here. Out of some leaves, consider only one leaf whose half portion, okay, the frontal half portion is inside the conical flax, like this here. You can see there in the diagram, which is there in the book also. So half of its portion is inside the conical flax. It is inserted into this conical flax containing potassium hydroxide. See here, this conical flax is already containing a chemical called potassium hydroxide, which can absorb carbon dioxide. So that if any carbon dioxide is there in the conical flax, is already absorbed by potassium hydroxide. That means there is no carbon dioxide at all in this conical flax. So this half portion of this leaf is inserted into this conical flax through this split cord. Here is a cord which is a split cord. Through this split region, this half portion of this leaf is inserted into this conical flax, where there is absolutely free of carbon dioxide, where no carbon dioxide. Alright, then what you do is that you keep this setup outside in the sun for some hours. Okay, so after some hours, you take this leaf out and again take it for the taste. Okay, starch taste. So you'll taste it again in the same manner. Alright, so in the taste, what you find there is that only the portion which is inside the conical flax where carbon dioxide was not there, you find turning to brown coloration. That means their photosynthesis has not taken place. That means there the photosynthesis could not take place there inside because of the absence of carbon dioxide. There was no carbon dioxide, so that photosynthesis could not take place inside, which is proved by this change in the coloration to brown. That means it is turning to brown, which shows that there is no starch. But where is the other portion of the leaf which was exposed to the atmosphere? You see turning to blue-black, showing that the presence of starch. That means in this region the photosynthesis had taken place. Okay, so from this experiment also what we can understand is that carbon dioxide is very much necessary for photosynthesis. Now we have the last experiment number 4 to show that oxygen is produced during photosynthesis. Okay, as you all know that photosynthesis is the one which not only produces or manufactures food but also releases oxygen to the atmosphere. Alright, okay, so for that to prove that, what you do is that you just take some aquatic plants growing in water, like hydrilla. I've taken hydrilla here, hydrilla plants. Take some hydrilla plants here in the beaker containing pond water. This is the beaker which has contained pond water. So there you take some hydrilla plants and cover it with a short-stemmed funnel. As you see here, the funnel, short-stemmed funnel. So you cover this hydrilla plants with this short-stemmed funnel in this manner. Alright. Then what you do is that take a beaker, take a test tube. Okay, take a test tube full of water. This test tube is there, full of water. Then invert it like this. Invert it over this short-stemmed funnel. Okay, as you see here, Okay, invert this test tube full of water over this short stem funnel and make sure that the water level in this beaker is above this stem of this funnel. Alright? Then keep this apparatus or keep this setup outside in the sun for some hours so that the photosynthesis continues there. Okay, so after some hours you see the gas out here. Okay, you need to go for the taste of the gas collected in the bottom of this test tube. Gas is there in its top as is shown there in the diagram, but which gas you need to taste it. We know that this must be oxygen, but you need to confirm it by testing it. So you need to test it by bringing this test tube close to the glowing splinter. Okay, so after some hours, when you are sure that the oxygen has been pumped out here, you remove this test tube very carefully. As soon as the water is dropped, you need to cover it with the thumb. Make sure that the gas is still there. Okay, and bring close to the glowing splinter. Okay, when you open this thumb, when you open the mouth of this test tube close to the glowing splinter, the glowing splinter now bursts into flame, confirming that the gas to be oxygen, because oxygen is the gas which support combustion. Okay, that's the reason why the glowing splinter bursts into flame. So from this experiment, what we understand is that oxygen is produced during photosynthesis. Okay, so that's all about the different experiments on photosynthesis. Alright, so in the next session, we'll try to finish up the chapter. Alright, so I hope you people have understood, and please go through the video again and again so that you understand to the fullest. Alright, so thank you very much.